All right, good morning. Today you are remote. There's no Zoom, so you can just work on this at home when you have a chance to do it. What you're going to do is, and I showed it to you in class yesterday real briefly, but you're going to simulate protein synthesis. So yesterday when we were in class, we looked at tissues, and you saw that the cells are not the same in every tissue, but we know that every cell has the same DNA. And so what we're looking at now is how that DNA actually is going to be used to make proteins. So the proteins do the work of the cells, and each cell, like you saw, had different jobs. So like the paramecium, all the things in the paramecium cell have to work together to make that one cell live as the organism in whole. Whereas skeletal muscle was more like rubber bands, where you had materials moving back and forth. So the structure was very different because it's involved in that movement. Whereas if I move my arm out, my skeletal muscles stretch in my bicep, and then when I pull it up, my skeletal muscles are contracted in my biceps. So that's why we saw those fibers sitting next to each other, which were the cells. Versus the stem of the plant, where they were very congregated and all condensed for support. Plus they had the clear structures, the cells that were clear for moving materials up and down. And then you also had the blood, where the cells were really scattered, because we know that the blood flows through the plasma to move materials through our body. So each of those cells have different proteins in them that do different work because they have different jobs. So what we're going to start to look at today is this idea of how these proteins are actually created from the DNA. And so you're going to create a fictional organism called a GNOPS. So when you go on to Google Classroom, this is the assignment that you're going to see. Please read through this knowledge probe. It gives you some good background information about DNA and genes. This should look familiar too. This is the structure of DNA with the copied nucleotides, A matching to T and G matching to C. Now, we know that DNA has to stay in the nucleus. Well, then how does the information get out of the nucleus and into the rest of the cell? So the way that that's going to happen is that it actually makes a structure called mRNA. RNA is very similar to DNA, and we'll take a look at it a little bit more tomorrow. But one of the differences is that there is a U in there instead of a T. So what that means is if the DNA has an A, like you see right here, the A bonds to a U instead of an A bonding to T. So this red line is this RNA. So everywhere where you see an A on the blue, which is the DNA, you see on the red that it's a U. And so this is how we can get that sequence of the DNA out of the nucleus. So the mRNA, which is complementary to the DNA, goes out, and it's actually read in groups of 3. So these groups of 3 are what are referred to as a codon, and they actually code for an amino acid. And amino acids then are used to make up proteins. This is what we're going to look at on Thursday and Friday in more detail. So don't worry about that too much yet. Okay, this is just to get some practice with actually seeing how DNA makes traits. So what you're going to do is the first thing you'll do is you'll click on the spinner. When you click on the spinner, you'll get this. Click and spin to get a number of which crossover you're going to use. So I got number 1. So then what I will do is I will go to the slideshow, and when I open my slideshow, I have this, okay? So depending on what number you roll will depend on what number of slide you go to. So I got number 1, so I'm going to go up here to slide number 1, okay? As I look at slide number 1, I can see that here's my DNA sequence for gene A, gene B, C, D, E, and F. So I'm going to take this gene sequence and I'm going to put it into my document. So I have gene sequence ACC, GGT, TAT for 1. So I'm going to come down into my document and I'm going to put, so I started it for you, so they all start with ACC, and then I had GGT, I believe, and then TAT. Oops. Okay. Or I could actually click on this and copy it and paste it. Okay. So either way, whatever works for you. Once you've got your DNA, you're then going to get your mRNA and then you'll do the amino acid and then the trait. And so this is an example down here of how to do that. So if I have ACC, remember that A bonds to U, C bonds to G, and then I would do the same thing here. G bonds to C, T bonds to A, okay? Oops. And then I would have my A, and then A is going to bond to U, and then T is going to bond to A, okay? So now I've got my mRNA. Remember that the mRNA then is going to be used to make the amino acids. So from there, I'm going to come down here. So I've got UGG, so my first amino acid is going to be 20. So up here I would put my 20. And then I have CCA. So now I'm going to find CCA, which is 12. And then I have AUA. And so AUA is 13. Okay, so I'm going to put my 13 here, like that. And if you wanted to, you could do like 2 copies of this document and then have a split screen so you can kind of see the both side by side. And that would be perfectly fine. My final thing is I have to figure out what's my trait. So 20, 12, 13. I'm going to come down into my amino acid chart. 20, 12, 13, hairy. All right, so my organism that I'm creating is going to be hairy. I would then do the same thing for each of these other genes. And then once I have all of my traits, I would come down underneath here. I'm going to draw it. Okay, so I would take out a piece of paper, draw it. If you want to use Google Draw, that's fine. It's up to you. You can just draw it on a piece of paper. It's probably the fastest. Take a picture and upload it into my, either share it, email it to myself, upload it into my Drive, email, copy it somewhere where I can then copy it, or just take my computer, take a picture of my organism, and then I'm going to copy that image here. I'm also going to click slideshow so that when I click that, I can then put my image here with my name. So I'm going to type my name, put my image, and then I will have my organism. Okay, and then we'll answer these questions here. You'll need to skip number 1. Okay, so you'll skip the question where you're going to compare it to your classmates until we have all of those. We'll do that in class on Thursday. But you do need to answer questions 2 and 3, which is what would happen if you were able to change your DNA, what might happen to your CHNOPS, and then also what would the DNA sequence be if it was plump. So you're going to work backwards, start with the trait, get the amino acids, get the mRNA, to then get the DNA. All right, so that's what you guys are going to work on today, and let me know if you have questions. You can always shoot me an email, and I hope you have a great day.